Welcome to Lecture 15. Sorry about that hurried end to the previous lecture. I don't know how loud that train sounded to you guys, but it sounded very loud to me. So, we're back to talking about meiosis. Just to make sure everyone got the very end of the last lecture, in prophase 1, there's a note on your lecture outline that both synapses and crossing over occur during prophase 1. Synapsis, again, then being the paired homologous chromosomes coming together, synapsing, and crossing over those homologous chromosomes, then trading pieces, trading segments, and each segment that's exchanged will contain a number of individual genes. And so by crossing over, we get new combinations of genes on each individual chromosome. We get new combinations of genes that we couldn't have had without this crossing over process. You'll notice now on your lecture outline that for both meiosis 1 and then for meiosis 2 also, if you flip over to the next page, we're talking a bit about the various phases of meiosis 1 and the various phases of meiosis 2. So prophase 1, metaphase, anaphase, telophase 1, and then on the next page, prophase 2, metaphase 2, anaphase 2, telophase 2. And you'll notice that these individual phases aren't diagrammed in your textbook, but they are shown and talked about very well in that second video. And so, again, kudos to the Amoeba sisters. Okay, so back to your lecture outline then. We're in meiosis 1 and the phases of meiosis 1. We've talked about prophase 1. Now metaphase 1. And here you might want to jot down in your notes that during metaphase 1, 2 lines of homologous chromosomes form. Metaphase 1, again, 2 lines of homologous chromosomes form within each cell. And if you think back to metaphase of mitosis, same idea. So anytime we have metaphase going on, whether it's in mitosis or meiosis 1 or meiosis 2, metaphase again implies chromosomes lining up along the equator of a cell. Notice then that metaphase 1 is followed by anaphase 1. And if you think back to mitosis again, notice you're seeing the same names for these phases now in the same order. So it's always going to be prophase, followed by metaphase, followed by anaphase, followed by telophase. And again, the same types of things are happening in each of these phases. And so anaphase 1 then, and during anaphase 1 of meiosis, homologous chromosomes move away from one another. Homologous chromosomes during anaphase 1 move away from one another. And then finally recall that telophase always results in the production of 2 daughter cells. And so at the end of telophase 1, at the end then of meiosis 1, we have those 2 daughter cells that we were talking about in figure 22.1 on page 826. And it's probably worth taking a minute just to go back and look at that figure again. So 22.1 on 826, again starting at the left, a diploid cell with 46 chromosomes, each containing 2 sister chromatids. And then remember, first meiotic division, so that's another way of saying meiosis 1, and the 2 arrows then coming to 2 identical cells in terms of genetics, and at the end of meiosis 1, then at the end of that first meiotic division, now again we have 2 daughter cells, as we said, each one haploid and each containing chromosomes consisting of 2 sister chromatids. So we are now halfway through meiosis, and we're ready to go back to your lecture outline and take a look at the second meiotic division. And again, second meiotic division, just another way of saying meiosis 2. And as we talked about then, when we initially went over figure 22.1, following along now on your lecture outline, and so meiosis 2 follows meiosis 1. Each cell that undergoes meiosis 2, so each of those cells formed by meiosis 1, each cell that undergoes meiosis 2 produces 2 cells, each of which is haploid, and so we're going from haploid cells to haploid cells. But notice now, at the end of meiosis 2, each chromosome consists of only one of those 2 sister chromatids. Again, as we said, talking about figure 22.1, very right-hand portion of figure 22.1, and the little note down in the bottom right, 23 chromosomes, so for haploid cells here, each chromatid, each sister chromatid, now an independent chromosome. And so let's run through the phases of meiosis 2, back to your handout, and talk about how that happens. So again, meiosis 2, it's going to be the same order, prophase, metaphase, anaphase, telophase, and now we just use the Roman numeral 2 after each one. And let me point out here, and you might want to jot this down, the phases of meiosis 2 are identical to the phases of mitosis. The phases of meiosis 2, again, are identical to the phases of meiosis, except, and you probably expected, well, there must be an except, so the phases of meiosis 2, again, are identical to the phases of mitosis, except with meiosis 2, we have a haploid cell producing 2 haploid cells. So again, at the end of meiosis 2, actually during meiosis 2, we're going from a haploid cell that we start with, and that haploid cell during meiosis 2 will produce 2 haploid daughter cells. Let's talk now about these 4 major sources of genetic diversity. Well, actually, let's go back to the phases of meiosis 2 for just a minute. And even though you're familiar with these from studying mitosis in A&P 1, let's just quickly run through the phases of meiosis 2. And so prophase 1, and if you just want to jot down here in your notes that the chromosomes become visible during prophase 1, the chromosomes become visible with an ordinary compound light microscope. So prophase 1, again, at the end of this process, the chromosomes have now become visible with a normal compound light microscope. And if I said prophase 1, and I'm not sure if I did, but again, prophase 2 we're talking about. So prophase 2, again, the individual chromosomes become visible with a regular plain old compound light microscope like the ones we have in our lab. Remember then, prophase is always followed by metaphase, and metaphase always involves chromosomes lining up along the cell's equator. Prophase, again, always followed by metaphase. Metaphase always involves chromosomes lining up along a cell's equator. And if you think about it, during meiosis 2, we no longer have homologous chromosomes. Because those homologous chromosomes, each pair of homologous chromosomes, were partitioned out during meiosis 1. And so during meiosis 1, then, if you think about it, during metaphase 1, we had 2 lines of homologous chromosomes, and we ended up then at the end of meiosis 1 with one of each of those homologous chromosomes going to one daughter cell and the other homologous chromosome going to the other daughter cell. But now, as a result of that, in meiosis 2, we don't have homologous chromosomes to work with, we just have single chromosomes to work with. And we get, during metaphase 2, a single line of chromosomes. And recall, at this point, each chromosome still consists of 2 sister chromatids. Each chromosome at this point consists of 2 sister chromatids that are still joined to one another. Anaphase 2 then, same thing happening as with anaphase of mitosis. So anaphase 2 of meiosis, the sister chromatids break apart from one another. Each sister chromatid is now called a chromosome. And the chromosomes move away from one another. And so again, very much like what happens with mitosis. And then finally, anaphase, remember, is always followed by telophase. So anaphase 2 followed by telophase 2. And recall, again, at the end of telophase, we now have produced 2 cells from each cell that we started with. And so during meiosis, we started with a single cell. Meiosis 1 gave us 2 cells, and then meiosis 2 gave us 2 cells from each of these 2. And so we go during the whole process from a single diploid cell to 4 haploid cells. And that is it for lecture 15.